Yeah, good afternoon, everyone, and thanks to the organizers for having me here. So today I'll be talking about my work on, or our group's work on non-coding variant interpretation using deep learning and evolutionary conservation as well. So in the first part of the talk, I'll introduce our new model, Promoter AI, which was developed by our colleagues Kishore, Nicole, and Gurman. That's going to be published in Science soon. And in the second part of the talk, I'll talk about some of our own work on expanding these methods to other non-coding regions, basically. So what's Promoter AI? It's basically a deep learning model that's used to predict impact of genetic promoter variations on gene expression. So basically, it can tell you if a variant would cause underexpression or overexpression or no change to gene expression. And the way these models are trained, we have two phases of training. The first phase is quite similar to what the models in the field do, like Informer and Borzoi. So the way it works is you have about 20 kilobases of sequence, and then you predict a bunch of these biochemical assay data sets like histone modifications, accessibility, transcription factor binding, and cage, which is basically just RNA data. It varies in two different ways. So the first thing is that we do all these predictions in a per base resolution. So some of the models like Informer, they do a 128 base resolution prediction. And we also focus only on the promoter plus minus few kilobases region, basically. So we don't train it genome-wide, we just train it centered at promoters. And really the biggest contribution in this line of work is the kind of second stage, or the fine-tuning stage. So this is, again, in line with a lot of how AI models are trained these days. You know, you have a huge pre-training step, and then you have a post-training or a fine-tuning step with higher quality data. So in our case, we've basically curated a set of promoter variants with known expression effects. And the way this works is in this kind of Siamese fashion, where you have the model replicated 2 times, and they share the weights, and you have one of the twin basically processing the reference allele, and the other twin processing the alternate allele. So the sequence is still 20 kilobases, but one nucleotide change. And then you take all the assay predictions, just average them, and then subtract them. And then you train this score to be close to what you've curated, basically. So some intuition on why this works is usually these gene expression models pick out on a lot of sequence biases like GC content and stuff, which is super correlated with histone modifications and whatnot. So when you have 2 identical sequences, almost identical sequences, you kind of encourage the model to pick out more causal features as opposed to correlated features. So how do we curate our fine-tuning data set? So for that, we look at GTEx, paired whole genome sequencing, and RNA-seq data from multiple tissues. And we look for the co-occurrence of 2 events. So the first event is a human must have a singleton promoter variant, and that gene must also have outlier expression across multiple tissues. So we call these outliers, and that's the outlier detection pipeline. So there's been a lot of methods in this field. And really the quality of fine-tuning, just like in like training ChatGPT, it really depends on how clean your fine-tuning data set is. So we want to quantify our purity as well. So in order to do that, we implemented a bunch of correction steps, like correcting for cis-EQTLs and trans factors. And also, like, the way we say that the data set is pure is by kind of setting the parameters of this outlier detection pipeline such that we can detect like 1,000 or so outliers by chance. But then when you pass the real data, you'll know how pure your actual data set is. So as you can see, if you just correct it for like genetic principal components and expression principal components, you'd get like 2,500 outliers for every 1,000 background outliers. But once you apply the cis-EQTL correction, which is basically just regressing out any kind of like contribution due to cis-EQTL. So we were interested in looking at rare variants, rare promoter variants. So these cis-EQTLs are very interesting in their own right, but not quite interesting for this task. So we want to kind of regress out those effects. We also remove the effects from trans factors. What I mean by that is, the expression value that you observe could be a function of expression of genes on other chromosomes. So we kind of like build a simple model to correct away these factors as well. So once you do that, you'll get around 4,000 outlier variants, which is 75% pure. Just to give a sense, like even models like ChatGPT are trained on somewhat this kind of purity. So we don't need anything more than this to train deep learning models. They're very noise resilient. So the next step we want to do is kind of like benchmark this very thoroughly. So I think this has been one of the biggest challenges in non-coding, is that we lack a lot of standard benchmarking suites. So we basically built this ourselves. And And we basically collected 6 different data sets. So the point is that in each data set, we get a bunch of variants that are known to cause underexpression, known to cause overexpression, and known to not affect the gene expression. So you can run a bunch of statistical tests on the RNA-seq or whatever the data is to kind of understand the actual effect, and then use these to benchmark your models. So because we have these 3 different classes of variants, we build 3 two-way classification problems, and then just go ahead and benchmark all approaches that we can see. That's been published. So the first data set is the KG5 saturation benchmarks. I think someone spoke about this yesterday as well. And basically, this is a MPRA data set which has been done on 10 disease promoters. So we also have another saturation mutagenesis benchmark, but this is a slightly bigger data set with 50 genes. It's yet to be published, but it will be published soon. The third one is MPRA on EQTLs and GWAS variants. The purpose of this MPRA was to kind of discern what's causal and what's just in linkage, basically. So, and then we can use that to benchmark our models as well. And in the second row, you see the GTEx EQTL benchmark. So this is basically fine map mutations from the official GTEx data set. And then we also look at proteomics data from UK Biobank. So there's like about 50,000 plasma protein measurements in UK Biobank, and you can kind of think that aberrant mRNA expression would also lead to aberrant like protein changes. So that's another way to evaluate our models. And then finally, we have RNA-seq data from Genomics England for about 5,000 participants with whole blood, basically. This is whole blood RNA-seq data. So that's another benchmark for us. First, I'll note that all of these are unseen. So none of these variants were seen during fine-tuning, or even these genes were not seen ever by the model. So this is complete generalization to new sort of tasks. And the way to read these plots is the darker the row, the better the model. So you can see that promoter A does pretty well, substantially better than almost all the published models as of today. So I just want to dissect like what's causing this performance gap. So first, if you look at a model like Puffin, this is a model that got released recently, and it was only trained on expression data. So they train on cage, rampage, and such RNA, just nascent RNA tracks. And they don't train on chromatin tracks, for example. So you can see that while that seems intuitive to do, it looks like you still need all these chromatin tracks to kind of pinpoint causal features as opposed to just correlations. But nevertheless, it's still pretty good, but it's just not as good as what you can do with all the tracks. The other side of this coin is ChromeBPNet. This is from Professor Anshul Kundaje's lab. It's a preprint. And this is trained only on chromatin accessibility data and not on gene expression. So this is kind of like the other side of the coin, and this is really good at MPRA benchmarks, but not so good at RNA-style kind of benchmarks that we have. So it tells me that you need to have both the RNA and the chromatin tracks to get optimal performance, which is what Basenji, Informer, and Borsoi do. So they train on all these existing kind of like assay tracks, and they're probably the second most competitive style of models, and they're pretty competitive in some other benchmarks, some of the benchmarks that we have. So the other thing I want to mention was I think there has been there are too many genomic foundation models these days, and we wanted to just try it out ourselves as well. So we independently benchmarked it 3 different times, and we saw we saw that they are not as competitive as what you would get with these kind of classical sequence to function models. I mean, they are still like 0-shot prediction, so we still maybe there is still room for improvement. But as of today, it's not easy to interpret variance with these data sets. But looks like there have been a lot of talks that have been improving these models. So hopefully there is scope in the future. Okay. Oh, so then to give you some intuition on what this fine-tuning process does. So that's kind of the meat of what this work does. We already know that these sequence-to-function models learn these TF motifs, and the way we know that is we draw these ISM plots, and we basically assign a per-base importance score to each of these bases. And then you'll see that before fine-tuning, this model picks 2 ETS motifs and a YY1 motif. Again, these are ETS, just multiple TFs could bind them, but we don't know exactly which one binds them, but some ETS would be binding it. But after fine-tuning, you'll see that an additional YY1 motif, it's in the reverse complement direction, has been identified by the fine-tuned model, which was missed originally. And not just that, you'll also notice that the ETS1 has also flipped direction, basically. While I acknowledge that there are tissue-specific effects, like the way these TFs work could be different depending on the tissue, but you can think of this as kind of a tissue-averaged effect of this TF. So basically, you'll see that once you fine-tune, you'll kind of learn the direction of these TFs as well more accurately. And to show that this is not a one-off just vignette, it's also like true for almost all the TFs that we have in the data sets that we benchmark. And you'll see that after fine-tuning for almost all of these TFs, you'll see a better accuracy after fine-tuning. So that's why the models are better at predicting expression. Then I want to like switch gears and just talk about like how to apply these models on kind of large-scale cohorts, like genetic and clinical cohorts. So the first one is UK Biobank. We know that it's like a data set of 500,000 whole genomes and like rich phenotype data as well. So we want to like, and then it also has like 50,000 plasma protein measurements. So we already showed that the model is very good at picking outliers in UK Biobank, but it also turns out that it's pretty good at correlating with the actual protein levels themselves. It's like you get a correlation of 0.5. So in that first plot, what you're seeing is on the y-axis is a z-score, which is just the protein level, and the x-axis is for that variant what the promoter AA score is. So you can see a decent correlation between the two, and this is across around 1,700 variants. So I said that UK Biobank also has thousands of phenotypes, and many of them are very correlated to each other. So you need to kind of cluster them, and you'll get about 40 to 50 traits, and about 100 genes that are linked to these traits. That was done through burden testing by some of our lab members. This was all published in 2023 as part of the Primate AI 3D work. So basically, we have kind of like gene to phenotype links that were kind of verified using PTV burden tests. So one such, I guess, trait is this alanine aminotransferase. So ALT is a very important clinical biomarker. So if you've ever done a liver function test, you'll get these values, and they're usually, super relevant. And you'll see that the promoter of the GPT gene, so GPT codes for this enzyme. So you'll see that the promoter A scores for variants in the GPT gene correlate quite well with the alanine aminotransferase levels in UK Biobank. So this is so the interesting thing here is that the model has never seen protein levels while training. So it's a good generalization across more complex kind of measurements. And to take it a step further, we also show that if you look at HDL cholesterol levels and look at LCAT and CETP gene promoters, you'll see a fair correlation between promoter A scores in these promoters and the HDL cholesterol levels. So this is just to show the impact of understanding variants outside of the coding genes, and hopefully this will get more people to be sequenced. So then after like common complex traits, we also next look at rare disease cohorts. So for this, we look at the Genomics England dataset, which is a very wonderful resource for these kind of analysis. Again, I think Illumina is very interested in this because today when someone with rare disease gets their genome sequenced, they basically don't get a really good answer unless they have some PTV in a very well-known gene. So we want to see what we can do about this diagnostic yield issue with kind of our variant interpretation methods. So in GEL, we focus on a set of 8,000 cases which are known to be monogenic. And we have experts who've curated like panels for each of these disease. Hopefully, these are the genes that create that phenotype that's observed. So we have about 100 panels or so on an average for most of these disorders. So what we do is we take the promoter variants from these cases, and then a singleton promoter variants from these cases, and just code them with the model, and then look at how often can promoter AI prioritize these panel gene mutations over like a background of other disease genes. So this tells you how likely is like a prioritized mutation to be inside the panel gene. You'll see that promoter A gets about like 1.5-ish enrichment. There's definitely room for improvement, but this still lets you do these kind of diagnostic yield analysis. So what you do here is you basically run this enrichment test for all different kinds of variants. For instance, if you do it with PTVs, you'll basically check if there are PTV variants within these panel genes, and you'll see that 50% of the cases can be explained with PTVs. I mean, this is 50% of diagnosed cases, basically. But you can also apply a lot of these AI methods. So there's still a lot of cases that are unsolved. So you can just see if there are Primate AI 3D prioritized mutations, missense mutations, or Splice AI prioritized, like exon intron, like splicing altering mutations, or Promoter AI prioritized promoter mutations. You'll actually see that you can kind of double how many cases you can solve once you apply these AI interpretation tools to these kind of large-scale disease cohorts. So that's good news for the community. So then we also looked at ClinVar. So again, I know that ClinVar only has missense mutations. There are some non-coding mutations, but they are all splice mutations mostly. So we get rid of all of those, and we are left with like 50 of them in like handful of genes. So that's why we don't show this as a main benchmark, but we just show this as a supplement where you have 50 pathogenic mutations from 10 genes. You can sample benigns. There are some benign mutations, but where we don't have benign mutations, we use like allele frequencies in populations to get benign mutations. You can see that as you get better at predicting expression, you get better at predicting pathogenicity too in the right direction. And we have a bunch of these examples, like 10 of them or so, and I'm just showing one of these examples. This is the promoter of this APC gene. So you'll see there is a YY1 motif in the promoter, and there are 4 ClinVar annotated pathogenic variants. You see that the model is also thinking that this particular motif is extremely important, and mutations in these motifs can create underexpression. You can also see that conservation kind of checks out really well with the motif. You can see that the conservation peaks at the motif and then dies down at the flanks. And you'll also see that the ChIP-seq evidence for this protein actually binding there is quite high as well. There's a clear peak at this promoter across different tissues. So this is just to show that you can use these models in conjunction with other methods like conservation and biochemical assays to increase your chance of interpreting the variant correctly. So then I'll just switch gears a bit, and how do we kind of move this to other regions of the genome? And we're interested in this because so what we call as promoters is a very small window around the TSS. So that's not it's a significant portion of the genome, but we still have a lot more left in the genome left to be interpreted. And as I showed, you can use conservation and these assay data to kind of use these synergistically to interpret variants in a lot of these regions. So towards this, we went out and collected a bunch of primate genomes. We have around 800 samples from 200 different species. Most of these species have been newly sequenced as part of this project. And this was done in collaboration with a bunch of labs worldwide, and this was all done by Lucas, Jacob, and Sabrina in their paper that published last year. So basically, what we show is that primates offer a very good kind of like a sweet spot where there is good alignment with humans, and there's enough variation that you can meaningfully compute some kind of constraint. So as you can see in this MSA plot, most of the human chromosomes align quite well with more than half of the primates that we have in our cohort. And we can use this to kind of call constraint bases, and then you'll see that more than half of the promoters and enhancers have some meaningful proportion of bases that are under selective constraint. Again, like the percentage depends on the exact promoter and like what it's actually doing, but you'll see that more than half of them have something going on that warrants some study. Yeah, again, I said that you can use all these different assays to have higher confidence interpretation, but the reality is that in the non-coding space, these signals don't often align very well, like the biochemical assays and the conservation scores. They don't align as well as they align in the kind of missense regime where, if you have something evolutionary constrained, it's likely to affect structure or something like that. So the reason here is, not all TF binding causes expression change, and not all expression change is super deleterious. So there's a lot of leeway for evolution to let neutral mutations just live, or even slightly deleterious mutations to live. But there are still enough examples that we can systematically tease these out. So I'm just showing one example from what we've been looking at. So this is the LDLR promoter sequence. So LDL receptor is a super important gene, super implicated in the cholesterol pathway, and very important gene altogether. And you'll see that if you just zoom into the promoter sequence, you'll see this TCA-CCCCA chimer that corresponds to a sterol regulatory binding protein DNA binding site. So this SREBF1 binds to a lot of genes in the cholesterol pathway, and again, this is a very important gene as well. So you'll see that if you look at the SREBF1 ChIP-seq data in HepG2 cells, you'll see that this locus has a very good peak. You'll also see that this region is open chromatin in liver, so that's all checking out. You'll also see that the phylo-P that you compute with mammal and primate data is pretty concordant. There is a peak, although much noisier, it's still concordant with what you observe with this data. But even more interestingly, if you look at the chimer and then just inspect the actual multiple sequence alignment, you'll see that this TCA-CCCAC is super preserved across the whole phylogenetic tree. So even though the flanking nucleotides might get deleted or changed to something else, you will just see that this particular thing is super conserved, because you can think of this as a equivalent of a domain in a protein, but this is in the regulatory regions. So basically cutting this off means that the gene never gets expressed, and hence the gene doesn't work. So it's super important to preserve this. And finally, I want to just talk a bit about what these constraint loci do and what kind of variants are enriched within this. So to do this analysis, we looked at UK Biobank fine mapping data for about 96 quantitative traits. And we also looked at the GTEx EQTL data set that we showed before. And what we do is we compute ratio of, or like an odds ratio of fine mapped to not fine map mutations within these annotations. And fine mapping can be thought of like pinpointing more likely causal mutations from just correlated but not causal mutations. So enriching for that means that you're likely to pick up something reasonable. You'll see that, and then in the x-axis, we kind of plot how deeply the element is constrained. So 65 million years ago, it represents primate constrained, but not mammal constrained. 100 would be mammal, but not vertebrate, and 160 would be vertebrate constrained, just super constrained. So as you can see, like as you get more and more constrained, these fine map variants tend to present themselves in these regions, which tells you that maybe they're doing something important, and that affects complex traits. And the reason even common mutations show some constraint is that over like long periods of time, even slightly deleterious mutations tend to get weeded out, and these they do more often than not hit constrained regions. So it doesn't work as well for EQTLs, as you can see in the first plot, but in the second plot, we've kind of stratified this into different constraint levels for the gene itself that the EQTL is talking to. You'll see that the trends are actually more concordant for EQTLs that are associated with constrained genes, and constrained genes here means just intolerance to loss of function. So that kind of makes sense why constrained regions would harbor these mutations. And finally, we show one more vignette. This is, I guess, like a HTLC variant. That seems to be altering a CEBP alpha motif, and again, as before, we also see the concordant evidence from different ChIP-seq and ATAC-seq and Phylopia and stuff like that. So I think, so we are very interested in this problem because if you go get your genome sequenced, you'll get a huge VCF file, and even in the protein coding space, you have mostly VUSS, and mostly non-coding mutations are presumed benign. But we are trying to show that they are not. That's not the case. There's it's a needle in the haystack problem, but still there is a needle in the haystack, so we want to go after it. And like broadly, the conclusions here is, you can use expression consequence prediction to predict pathogenicity better. You can build these foundation models using all sorts of accessibility and histone modifications and these RNA-seq data sets. You can fine-tune them if you have a curated set of well-curated set of expression outliers and perform even better. And we've also built benchmarks that span a lot of different assays and hopefully serve as a good resource for the community as well. And finally, we've been showing that even these constrained enhancers and constrained UTRs tend to have these expression-altering trait-associated variants in them. And yeah, I want to acknowledge like all my group members for all their work and help. And yeah, I'm happy to take any questions.